Okay, so it's a couple of minutes after the hour, so I think we're safe to say that most people are here, so we'll get going. So firstly, thanks for joining my webinar today. My name is Jen, and I'm a Senior Scientific Sales Executive with Rapid Novor. But today we'll be discussing how we've seen scientists fully harness the power of immunotherapy through protein sequencing. Now, before we officially kick things off, I would like to do a quick poll to see where everyone is at. And so the first question is, what are you hoping to get out of the webinar today? Do you want to learn more about immunotherapy or about protein sequencing, maybe see new technologies in immunotherapy, or all of the above? So I'll just give it a couple of seconds to everyone click their answer, and then we'll see what the results are. Okay, looks like most people have done the poll. So I'll share the results. So that's great. I was expecting a lot of people to say all of the above. So that's really awesome to hear that we have an enthusiastic group today. And nice to see that there's some people that want to learn about protein sequencing and also some new technology in immunotherapy. All right, so we'll get going. There we go. So to start things off, the role of the immune response is not a new or novel concept, as of course it plays a vital role in defending diseases such as cancer. And interestingly enough, all the way back to ancient Egypt, there are records showing tumor regression. BC China and early Europe have records showing these observations too. With the general concept of vaccination, this notion of using the immune response was known, but they didn't know what the immune response truly entailed. And back then, we just didn't have the tools or understanding to fully harness the power of the immune system. And the knowledge of the immune system in cancer would not come in till about the late 18th century, when two German clinicians made separate observations on tumor improvements in patients with concurrent superficial skin infections. A few years later, up to 1,000 inoperable cancer patients were successfully treated by inoculating them with heat-inactivated bacteria cultured from those skin lesions. Now, many discoveries, of course, during this time were made in parallel, where we really started to use the immune response to our advantage. It took a couple of centuries to really characterize the immune response, and much of the understanding is thanks to the discovery of antibodies, which happened in the 19th century. And thanks to this discovery, you can take inventory and identify the cells of the immune system. And so I hope with this presentation, I can show you how vital antibodies are in immunotherapy and some of the awesome applications that I personally have seen researchers use. And as I'm sure we're well familiar with, the immune response has three main components: intrinsic, innate, and adaptive immunity. Innate and adaptive are typically exploited in immunotherapy. Adaptive immunity is then subdivided into humoral and cell-mediated immunity. And it is with this close interplay in between these two responses where we can get an effective immune response. And since antibodies tell the story about what the immune system has been exposed to, these proteins are essential to cataloging that immune response. And with their unique nature of intrinsic binding properties, antibodies really do become essential weapons to fight pathogens and to recruit immune effectors. So in this presentation, we'll be going through some case studies to show how antibodies make a huge impact in immunotherapies. Without the discovery of antibodies, we wouldn't be able to properly catalog the immune response and its roles. And understanding this has allowed us to create a whole new area of disease treatment, which is harnessed through vaccines, antibodies, and cell therapy. And as I mentioned today, we'll be going through many case studies of immunotherapy applications that I've seen. We're going to break this into 3 sections. So treatment, which includes immune checkpoint inhibitors, T-cell transfer therapy, and virotherapy. Monitoring, and then research and detection, where things such as immunoreagents and biomarkers fall under. So for now, we'll quickly cover the types of immunotherapy, which include antibody-based, cell-based, and viroimmunotherapy. So I have another quick poll for you guys. And this one is, what area of immunotherapy is most interesting to you? Do you like the virus side of things, or antibodies, maybe cell therapy, or all of it? So I'll give it a couple more seconds for everyone to get their responses in. Okay, looks like most people have got them in. And here's the results. So yeah, it looks like we have a lot of antibody people in the crowd. So that's really great. And a lot of people love all the aspects of immunotherapy too. So that's really, really awesome to see. Okay, so first we'll get started with some antibody-based immunotherapy. And when people are looking to fight pathogens, you're typically targeting a certain antigen to tell your immune system to fight it. But typically when you're looking at cancer, you're probably targeting checkpoint inhibitors, which are proteins that modulate the cytotoxic T-cell response to dampen inflammation. Now, by blocking these checkpoint inhibitors, such as PD-1 or CTLA-4, you can continue the T-cell response and ensure the close interplay between humoral and cell-mediated immunity remains on. So T cells are activated when their TCR simultaneously engages to both the MHC presenting a specific antigen and the co-stimulatory receptor CD28. Now, aside from Treg cells, activated T cells also begin the process of displaying CTLA-4 on their surface. The latter ensures the late after activation, T cells have a way to regulate their cytotoxicity by permitting cell surface expressed CTLA-4 to bind to B7-1 and B7-2 molecules on APCs, and thereby preventing binding of CD28. So this inhibitory mechanism results in a lower activation state and the eventual assuagement of inflammation. Many cancer cells exploit this mechanism by promoting an immunosuppressed environment. And as scientists continue to innovate, I'm sure that more antibody types will be added to this chart, but this is just here to point out that it's amazing that we have such a wide variety of antibodies or antibody constructs to choose from and work with. And we've really designed quite a catalog and extensive toolkit for antibody immunotherapies from our studies of the immune system. Now, cell-based immunotherapy, also known as adoptive cell therapy or autologous cell therapy, is a well-established type of immunotherapy that has benefited from recent innovations, namely gene editing. So here, tumor-specific T cells are extracted from and infused back into the patient for targeted tumor killing. Cells are taken either from blood as PBMCs or as pluripotent stem cells from the bone marrow of the patient. Isolated T cells are genetically engineered via transduction, the introduction of a vector. So the gene that is inserted into the patient's T cell's genome is that of a chimeric antigen receptor, known as a CAR, a synthetic fusion of a MAB fragment known as the antigen recognition domain, and a TCR fragment, namely the TCR signaling domain. So cell-based immunotherapy is really the perfect marriage of humoral and cell-based responses to promote cell-based immunotherapy. A huge role is still played by antibodies here because their binding region is used, which makes the therapy specific to the target. Now, oncolytic viruses are viruses capable of destroying tumors through cell lysis. Of note, lysis of tumor cells results in the release of several factors that alert the immune response. Now, in 2015, the first oncolytic viral immunotherapy called TVEC was approved by the FDA for treatment of advanced melanoma. Now, TVEC's backbone is a lytic DNA virus herpes simplex 1 modified to express the cytokine acting molecule GM-CSF, which stimulates immune cell proliferation. So cells whose proliferation, migration, and survival mechanisms are unchecked are optimal for a virus, especially because as a result, these cells have lowered antiviral responses. Finally, the use of viruses in immunotherapy boasts the added bonus that they may incite an immune response. However, it's important to note that though viruses would naturally favor tumor cells, they also employ tactics developed throughout evolution that might enhance tumorigenesis. So therefore, engineering is required in this case. Now, one of the many ways these viruses are engineered is by encoding monoclonal antibodies to deliver mAb immunotherapy directly to cancer cells and thus bypassing potentially harmful off-target effects. So for example, a gene encoding a human anti-CTLA4 mAb has been successfully inserted into adenoviruses, which in turn has resulted in increased anti-tumor activity of T cells in cancer patients. Another attempt has been to synthesize oncolytic viruses expressing anti-PD-1 antibodies or antibody fragments, yielding promising results in several studies. So again, here we see the importance of fully understanding the antibodies that we're working with. And really, at the end of the day, the most important part of the antibody, at least we believe, is its primary amino acid sequence. That primary structure of the antibody has, also things like post-translational modifications. And of course, that exact sequence of the amino acids is absolutely crucial. In a 2017 study, we found that almost all CDR regions have isobaric residues, such as isoleucine and leucine. And when trying to extract that amino acid sequence, the binding can be affected if that sequence is incorrect. So as many people know, in the antibody space, glycosylations is also a huge area of interest. So it's really imperative to identify those. And there's also specific post-translational modifications associated with antibodies that are instrumental to their structural stability, their half-life, and their binding properties. To successfully engineer an antibody or a portion of it for its ligand properties to generate a potent immunotherapy, you really do need access to the protein sequence. Of course, monoclonal antibodies are vital to many aspects of immunotherapy, as we've covered, and their innate ability to bind antigens makes them indispensable weapons and tools in the fight against cancer and other diseases. With increasing immune evasion observed from cancers and viruses like HIV, it becomes even more pressing to discover and develop novel antibodies for more potent, consistently efficacious treatments. Current approaches to antibody discovery include hybridoma development, in addition to more recently established technologies such as next generation sequencing of B cells, as well as display technologies. Though incredibly useful in the development of mAb therapeutics to date, these methods do have some shortcomings that result in a lengthier process of discovery and development. So some of these limitations may include anything from loss of specificity or cross-reactivity to missing information or prolonged timelines, or simply just not being able to view the whole picture. And so what I'm presenting here today is a proteomics-based approach to discovery through next-generation protein sequencing. So a lot of you came here today to learn more about protein sequencing. So what exactly is it? And essentially at its basic level, We use a cocktail of enzymes to digest a protein into many fragments. These peptides are then analyzed using mass spectrometry and then reassembled using an algorithm and specialized software. I've heard people say that protein sequencing is ambiguous, expensive, and timely. And well, 20 years ago, yes, it was. However, recently, Rapid Novor has created a team to exclusively focus on protein sequencing. That's all we do. And we've created a platform that's highly accurate, robust, routine, and fast. Since we do everything in-house, you honestly won't find a better team of experts than our scientists at Rapid Novor. So now is some fun parts. I'd like to show you the applications of the technology to see exactly how protein sequencing is being implemented in immunotherapy pipelines. In this case study, we'll be looking at a recent patent published by Janssen. This patent is based on the premise that gamma delta T cells, which mainly express heterodimers of TRGV9 and V delta 2 chains, demonstrate potent anti-tumor functions. These cells express TCR TRGV9, and the majority, if not all of these cells, exhibit efficient cytotoxicity of tumor target cells. Now, this ability is then harnessed using bispecific antibodies constructed such that one arm binds to TRGV9 structure and the other arm binds to a second T cell tumor associated antigen expressed by the tumor cells. So therefore, the bispecific antibody bridges that effector and target cells together, resulting in tumor cell killing. So Janssen needed to modify the sequence into a bispecific, which of course was difficult to do without that amino acid blueprint. And they only had access to a protein sample, so they took advantage of our REMAB monoclonal sequencing platform here. Now, Janssen sequenced 2 antibodies with us for this patent, in which they used the sequences to engineer into the bispecific. So we obtained excellent results on the antibody sequence side due to the immense level of coverage you see here. It might be hard to see, but there's lots of tiny lines in this colorful area, and each line is a peptide overlapping. So, Janssen really trusts us to get the sequence of their antibodies correct the first time. And we're really confident here that we are sending them 100% accurate sequences every time, just due to the immense amount of evidence that we're getting. So, what we're looking for is a minimum of 30 times coverage over every single amino acid. And in most cases, just like the one here, we're seeing hundreds of times overlaps, which is why we are so sure of the sequences. Now, Janssen expressed and purified the anti-TCRV gamma 9 antibody, which demonstrated binding to human gamma delta T cells, showing that specificity towards TCRV gamma 9. So that was really great news for them. And from there, Janssen also sequenced an anti-TAA2 clone and constructed that anti-TRGV9, anti-TAA2 bispecific human IgG1 antibody. Now, this was then tested for T cell redirected killing of H929 cells, which express TAA2. This is just one of the figures from several in this patent that show that this bispecific did bind gamma delta T cells and mediate gamma delta T cell cytotoxicity against TAA2 expressing H292 cells in vitro. So we're really happy to see such great achievements come from our sequencing, and getting that sequence right was really critical to the success. And I have another invention to share with you guys, this time from Capienda Biotech. And here they disclose an engineered extracellular vesicle compromising of a fusion protein having petrospanin, or a functional fragment thereof, linked to an anti-TLR antibody that inhibits an endosomal TLR, and also a method of treating an inflammatory response in an individual in need. So here, anti-TLR antibody clones were selected and sequenced using our monoclonal antibody sequencing service. The scientists at Capienda were able to use the protein sequence to engineer the antibodies as the payload in the form of a single chain FV as fusion proteins with the tetraspanons. And this was done to generate prototype engineered exosomes for the invention drug delivery system. Now, the correct protein sequence here was really crucial to ensure a smooth and efficient engineering process for this invention. Now, as antibodies are natural ligands produced by immune cells with high specificity and affinity, they are regularly chosen as the main approach for targeted therapy. In targeted cancer therapy, antibodies or antibody fragments have been used as carriers for the delivery of a variety of anti-tumor molecules, such as siRNA, cytokines, drug-loaded nanoparticles, etc. Now, in the treatment of inflammatory disorders, antibodies conjugated with inflammation inhibitors to selectively target inflamed tissues have also been a promising therapeutic modality. This system takes advantage of the specific antigen binding ability of antibodies, along with other benefits, including extended half-life time and better tissue penetration. Now, as an emerging treatment strategy, targeted therapy is still under exploration and development, especially for the early stage selection of antibodies. In order to achieve precise targeting, the specificity and affinity of antibodies really does need to be strictly assessed so that healthy cells and tissues will not be targeted. Meanwhile, novel antibodies with appropriate functionality still need to be discovered and identified to increase the diversity of the drugs and types of diseases to target. Now, DECA Biosciences is a biotech specializing in next-generation targeted cytokine therapies, focusing on inflammatory diseases and oncology. Their passionate team of scientists have addressed all of these challenges associating with turning naturally occurring and modified cytokines into medicines. Their half-life extending tissue targeting enhanced manufacturing scaffold platform known as DiaKine enriches disease controlling dual cytokine combinations directly into affected tissues. This technology for enriching DiaKines in the tissue or cells of interest is a key differentiator for DECA. When creating targeted drugs, knowing the exact sequences of the antibodies of interest is critical for their drug design, development, and function, which was one of DECA's key challenges. Now, in order to engineer the right scaffolding system for each disease of interest, DECA's team required the full knowledge of their key antibodies. And due to this need, DECA's team decided to outsource their antibody protein sequencing to Rapid Novor. And using our REMAB de novo sequencing platform, we determined the amino acid sequence of DECA's antibodies, of course, without requiring access to cell line or DNA sequences. Now, using that sequence information, DECA has designed and developed a number of DiaKines, specifically engineered to suppress inflammation or to stimulate the immune system. The potency of these DiaKines is enhanced when they are targeted to the tissue or cells of interest. DECA has several pipeline dual cytokine candidates, which are currently in preclinical development. Now, we had another client use our service for viral immunotherapy applications, and they sequenced an antibody specific to what they wanted to target, which was then used in the virus. Now, as coming from the sales side of things, my favorite part of the job really is seeing all this amazing research emerge and new tactics that people are employing. We also work with a lot of scientists in the CAR T world who use antibody sequences to engineer that onto their CARs. Okay, so we've talked a lot about immunotherapy treatments, and now I'd like to go over disease monitoring. And the example that I have for you today is our very own technology that we've developed called EasyM. I wanted to highlight how knowing the protein sequence can be useful in augmenting the specificity and sensitivity of an assay. We've developed an assay to track the M protein of patients with multiple myeloma. The M protein is a monoclonal antibody or light chain often secreted abundantly by patients with multiple myeloma due to cancerous plasma cells. Now, with knowledge of the exact sequence of the antibody from each patient, we can easily follow disease progression more specifically and sensitively. We begin by first sequencing the patient's specific M protein, and then can monitor the unique patient sequences over time. Because we're able to specifically target the M protein, this allows us to be more sensitive than other traditional methods like SPEP or IFE and detect relapse much easier and in a non-invasive way. Our application of this technology in M protein monitoring highlights the importance of knowing the protein sequence of antibodies for a more refined targeting of a disease and, truly personalized medicine. So our last category for these applications is research and detection. So here's an example of how our tech can enhance both your research and production efforts. So since exosomes are shed by both normal cells and those under pathological conditions, they are considered crucial biomarkers for clinical diagnostics. Now, a company approached us with an antibody that they used to capture exosomes for a diagnostic test, and they were concerned about reproducibility of this antibody, but they did not have access to the cell line. So thanks to our REMAB monoclonal sequencing platform, they were able to obtain the full protein sequence with additional observations such as post-translational modifications. And you can see again, the depth of coverage that we're getting here is quite immense. Each tiny line on the figure is a peptide we found, and we're really getting tens and hundreds of overlaps over each individual amino acid. This is why we're so sure of the sequences that we provide. And so this group that used us found that the recombinant antibody worked much better. And with the recombinantly produced monoclonal, the antibody had higher sensitivity and therefore greatly improved the results of developing this diagnostic test. Okay, so this brings me to the point in our presentation where you might be wondering a little bit about how our technology actually works. And so I'll be going through that in our following slides. So to describe REMAB, I just like to break it down into 4 steps: enzymatic digestion, mass spec acquisitions, REMAB assembly, and final review by our bioinformatics experts. With this process, we're able to get people 100% accurate protein sequences in just 2 weeks. And that accuracy is really owed to our requirements for the amount of evidence needed to make a call on the sequence, which includes that minimum 30 times coverage over each individual amino acid. And the first thing we do when a sample arrives is check the quality by running a reducing SDS page gel. So this separates the heavy and light chain, and ideally we see 2 perfect bands like in this picture. However, we do see quite a lot of samples with contamination of other proteins such as BSA, HSA, etc. So if we see a sample like this, this is why our screening process really is important to us, because if we don't do this, then our mass spectrometers are just going to get crowded with contaminating protein peptides. So ideally, we get a sample and it's nice and clean and it doesn't need any cleanup, but sometimes we do have to run some columns to purify things out. Well, once that's done, we do have some chemistry to separate the heavy and light chains and cleave glycans. This material then gets digested by different enzymes, which will then cleave the protein at different positions along the amino acid chain. Now, we've chosen the enzymes very carefully so that they have very different cutting rules to increase digestion site diversity. These digestions are run on our LC-MS system to generate peptide data sets. And our Novor engine interprets the MS data and converts it into peptide sequences. The REMAB assembly algorithm then assembles the peptides into that final protein sequence. And of course, this is science, and there's always a curveball that science throws at us in anything we do. And in the mass spec world, the hurdle is isobaric residues or combinations of residues that equate to the same molecular mass. And the most common challenge here for us is isoleucine and leucine. This is actually one reason I've heard people say why they shouldn't do de novo sequencing due to this ambiguity of isobaric residues. However, this is simply untrue because there is a way to distinguish them and with 100% confidence, and we do it tens of hundreds of times a day or a week. And what we're doing is using the W-ion method, which was actually first described in literature maybe 30 years ago or so, but Rapid Novor was the first to commercialize this into an actual solution. So at a first glance, isoleucine and leucine have an identical molecular mass, 113 Daltons. So you'd think that they're indistinguishable on mass spec. But despite this, they do have unique R groups. And what we can do is fragment these individual amino acids further to obtain W ions. We then measure this loss in mass, and from that data, determine whether the residue is an isoleucine or leucine with unmatched accuracy. Now, traditional approaches might look for germline sequences or rely on the enzyme digestion rules, but neither are truly reliable or de novo. And when we regularly see 5 or more isoleucines or leucines in the CDRs, accuracy really becomes key here. So our goal at Rapid Novor is to deliver you a single sequence that just works. And that's why we've developed this technology. And of course, we're not going to give you a sequence just spit out of a computer. A huge part of this process of sequencing antibodies is bioinformatics review. And that's done by a minimum of 2 independent bioinformaticians. This ensures that the sequences are meeting our minimum quality standards, in addition to an array of all kinds of other checks. What is great about that is everything is in-house. So if there happens to, say, be a gap in the sequence due to a hydrophobic area that an enzyme couldn't cut into, the bioinformatics team can just send a message over to the lab to do an additional digest with a different enzyme, and then the lab can get started on it immediately. Now, this step of bioinformatics review is extremely critical and ensures that we obtain 100% accuracy and 100% coverage. So what's unique about us? First off, we're using the most sensitive mass spectrometers on the market and simply looking for an enormous depth of coverage and evidence. That 30 times overlap by individual peptides over each amino acid ensures that we are 100% confident in the sequence. Our wild technology allows us to have no guesswork or ambiguity in the sequence. And our software is also real-time sequencing, meaning that our mass spec scientists can monitor samples and sequences as they fly through the mass specs and make adjustments to the instrument settings along the way to obtain optimal data. Our independent review and accuracy standards are extremely critical, and honestly, the bioinformatics team won't accept anything less than 100% accuracy because they too are scientists and have been in the lab and know the struggles, and they really understand how important it is that the sequence is 100% correct. Okay, so to sum up the monoclonal sequencing service, we use that multi-enzyme digest and LC-MS acquisitions to sequence monoclonal antibodies accurately and quickly with full coverage of the heavy chains, light chains, CDRs, and including PTM insights. Rapid Novor is a team of experts in what we do. We've hyper-focused in the single area Sequencing is all we do. We live it and breathe it. We've sequenced close to 4,000 antibodies now, and we are proud to be Canada's largest privately funded proteomics facility. So we have a lot of mass specs. As for sample requirements, we don't need too much from you, actually. We just ask for 100 micrograms of antibody protein of interest at a minimum 80% purity. The best thing about our technology is that it's fully de novo, meaning that we don't use any kind of database to sequence. The results are based solely off the mass spec data. And so that's why we can sequence any species and any isotype. Okay, so today we covered REMAB, our monoclonal sequencing platform, but I should let you know that we do have REPAB, polyclonal sequencing technology, in which we can sequence monoclonal antibodies directly from a polyclonal sera mixture. In the future, I also hope to talk about more services, including Novor, which is useful for providing a relative quantification of proteotypic peptides of interest, which may be useful for biomarker work or vaccine studies. Additionally, MatchMab is a useful tool for confirming sequences of antibodies. And that concludes today's webinar. So thank you again all so much for attending. I really appreciate your presence and participation in some of the polls. Again, my name is Jen and my email is here on the slide if you have any questions or want to talk offline. And for now, I'll stay live for a few minutes to answer questions. If yours doesn't get answered, don't worry, I'll be sure to follow up via email. Okay, so we got a question. On average, how long does it take Rapid Novor to sequence MABs? So that really depends on the volume. If you send in one antibody, we can sequence it in just 2 weeks. We also have an expedited service option in which we can sequence it in just 1 week. If you're sending a batch up to 10 antibodies, we can get all of those done in 3 weeks. You know, if you send in 20, we can get that all done in 4 weeks, etc. So the process has been really streamlined to allow for high throughput sequencing of these antibodies, which it has been really great. And one more question says, can you do a conjugated antibody? So the answer is yes, we can sequence conjugated antibodies as long as that conjugate is done non-site specifically. So, you know, we see these things all the time, like PE or fluorophore labeled antibodies, and those are really no problem for us at all. Okay, so I think that kind of sums up all of the questions that we've gotten today. So again, thank you so much for attending this webinar. My email is there. The last name is pronounced Surha. Yeah, I really hope you had a great time, and thank you for joining.